welcome to everybody to the Science of Childhood Cancer. I'm Doug Green. I'm hosting today. Before we get started, let me remind everyone that you can ask questions by either putting them into the chat. The little thing that looks like a word bubble button will open the chat and you can type in your questions. You can also email your questions to SOCC, Science of Childhood Cancer, at stjude.org. We'll be monitoring those. Our speaker today will stop about halfway through and will entertain some of the questions then. I'll be reading them, but if you identify yourself, I will mention whose question it is. And then at the end, we will end at the top of the hour. We will leave this open for an additional 30 minutes if desired to continue a conversation and to ask as many of your questions as we can during that time. So plan to stick around if you can. If not, the seminar will end at the top of the hour. And we can get started. It is my very real pleasure to introduce my colleague and friend, Richard Kowacki. Richard received his PhD from Yale University, where he studied chemistry and biophysics. He then moved to the Scripps Institute in La Jolla, California, where he used NMR technology to probe the molecular mechanisms of cell cycle machinery. Richard was then recruited to St. Jude, where he was the first new recruit, junior recruit, to the, at that time, new Department of Structural Biology. Richard's research focuses on intrinsically disordered proteins. Now, they're intrinsically disordered. How can you do structural biology on them? But Richard has developed the methodologies and the approaches to use sophisticated biophysical and biochemical techniques to understand how unstructured proteins control structure and mechanism. More recently, he's moved into the field of phase transition, where he's made fundamental contributions to that emerging field, which I think is the topic today as we explore phase separation and gene transcription from the nucleolus to aberrant transcription in leukemia. Take it away, Richard.
Thanks very much, Doug. Can you hear me okay?
We can hear you fine, thanks.
Very good, thanks. So I first want to thank Drs. Green and Roberts for the opportunity to present our research in the area of phase separation to everyone today. And I want to welcome everyone to this lecture and thank you for attending. So in my lab, we use a variety of biophysical and structural methods to understand the conformational and dynamic features of biomolecules. And some of those techniques are illustrated on the right of the slide. And through these studies, we seek to understand the basis for intermolecular interactions that lead to the formation of intermolecular networks, as illustrated in the center of the slide, that underlie the process of liquid-liquid phase separation, which in vitro leads to the formation of condensates, as illustrated here, and to the formation of membraneless organelles or other condensates inside cells. And we seek to understand how the process of phase separation mediates biological processes and controls cellular behavior. And so today I'll begin with a brief introduction to the topic of phase separation in biology. Then I'll move on to nucleolar biology through the lens of phase separation. Then I'll close with phase separation by NUP98 fusion oncoproteins in leukemia. And as Doug said, I'll take a break after the nucleolar biology section to take some questions if there are any in the chat box. So I first want to acknowledge and thank a growing cast of coworkers. Those on the left were more long-standing members of the lab, Some have since departed, and some of the newer members are shown on the right. Unfortunately, I don't have time to acknowledge them individually, but I greatly appreciate their dedication and important contributions to the lab's research. Also want to acknowledge and thank a number of collaborators listed on the bottom left, and importantly, want to acknowledge sources of funding, the NIH, ALSAC, and various sources from St. Jude, as well as Rhodes College. So numerous membraneless organelles are known to exist in cells, and in the last decade or so, it's become understood that these form through the process of phase separation. So there are membraneless organelles in the cytosol as well as in the nucleus. So in the first part of the lecture today, I'll focus on the nucleolus. So the nucleolus is the largest membraneless organelle in a cell and the first described. So it was described more than a century and a half ago. Cancer cells, importantly, display enlarged and more numerous nucleoli, and this has been known since the late 1800s. The nucleolus is comprised of the genes that encode ribosomal RNA, the ribosomal RNAs themselves, as well as both ribosomal and non-ribosomal nucleolar proteins. Importantly, the nucleolus mediates ribosome biogenesis, but also is a center for cellular stress signaling. So first, I'd like to provide a brief introduction to features of the process of liquid-liquid phase separation. So this process is mediated or is driven by weak and transient interactions between multivalent biomolecules. It's exquisitely concentration, temperature, and ionic strength dependent, and can be modulated by post-translational modifications, making the process regulatable. It occurs through formation of transient non-covalent networks between biomolecules, as I illustrated in my introductory slide. And importantly, this process organizes biomolecules within cellular compartments to perform diverse biological functions. So today I'll discuss ribosome biogenesis and transcription and how these processes are mediated by phase separation. Just some terms before we get going. So shown here are droplets or condensates prepared in vitro. So I'll refer to these as droplets or the dense phase or condensates, and the surrounding solution I'll refer to as the solution or the light phase. So the proteins fibrillary and nucleophosmin organize the multiple layers of the nucleolus by undergoing phase separation with ribosomal components, both ribosomal RNA and ribosomal proteins. So a nucleolus is illustrated on the upper left. This is from Cliff Brangwen's early pioneering work that demonstrated that the nucleolus is in fact a phase-separated condensate, and that it exhibits liquid-like features. But the nucleolus is really cool because it exhibits multiple subcompartments. And so each of these green bodies has a multi-layered structure that's shown schematically in the center of the slide. So at the very center is the fibrillar center, which is comprised of the genes that encode the ribosomal RNA, so the rDNA. And transcription of ribosomal RNA occurs at the interface between this inner layer and the second layer that's called the dense fibrillar component, or DFC. So the transcribed RNA instantaneously phase separates with the protein fibrillary, creating this second layer of the nucleolus. And in this layer, the ribosomal RNA is processed. It's spliced and it's modified. And due to constant transcription, then there's a flux of ribosomal RNA from the inside to the outside of this layer then when the processed RNA exits the DFC, it instantaneously phase separates with the protein nucleophosmin, which captures the ribosomal RNA in this third and outer layer of the nucleolus. But nucleophosmin not only phase separates with ribosomal RNA, but it also phase separates with ribosomal proteins. So ribosomal proteins, of course, are synthesized in the cytosol. They're transported through the nuclear pore into the nucleoplasm, where they're then sequestered through phase separation with nucleophosmin, creating essentially an incubator for assembly of ribosomal subunits. So the ribosomal RNAs and the ribosomal proteins then encounter each other and assemble into ribosomal subunits in this third granular component layer of the nucleolus. So how do the two layers, two outer layers of the nucleolus sort of stay separate from each other? So in a series of studies done together with Cliff Brangwyn and Rohit Papu, we showed that fibrillary in vitro can phase separate with ribosomal RNA, as can nucleophosmin. And key observations were that the fibrillary droplets were viscoelastic with high surface tension. In contrast, the nucleophosmin droplets were highly fluid with low surface tension. In addition to having different surface tensions, these two fluids were shown to be immiscible. So the laws of fluid physics then dictate that based upon the rank order of surface tensions for immiscible fluids, they can take on different sort of arrangements, in some cases having multi-phase droplets or two independent droplets. So the rank order of the surface tensions of the fibrillary and nucleophosmin droplets with ribosomal RNA fulfill this condition, which dictates that the fibrillary droplets, due to their high surface tension, will exist within a surrounding droplet formed by the nucleophosmin with ribosomal RNA. So essentially you have two immiscible liquids arranged as illustrated here. And so in this report from a few years ago, we demonstrated that in fact this arrangement of these two types of droplets was recapitulated in vitro phase separation experiments, and that these differences in surface tension between the two layers explain why the DFC resides inside the granular component of the nucleolus. So the key take home is that the fibrillary-rich DFC exists within the nucleophosmin-rich granular component due to differences in surface tension between the two layers. It's really as simple as that. It's just rooted in fluid physics. And in addition to having greater surface tension, the DFC is more viscous than the granular component. And we think that this will, of course, affect the diffusion of molecules in these two layers. And the higher viscosity of the DFC may serve as a timer controlling the resonance time of ribosomal RNA in that compartment to allow for its full processing before diffusing into the granular component. So just to summarize what I've just told you, this multi-layered structure of the nucleolus allows for ribosomal RNA to be transcribed in the fibrillar center, in the center of this hierarchical layering, and is then engulfed by fibrillary, creating the dense fibrillar component, again through phase separation. The ribosomal RNA is processed in the DFC and next migrates to the surrounding granular component. So in the GC, through phase separation, nucleophosmin mediates the assembly of ribosomal RNA with ribosomal proteins to form pre-ribosomal particles. It's really an amazing assembly line with different aspects of the overall processes of ribosome biogenesis compartmentalized through phase separation. So what role does nucleophosmin play in the granular component of the nucleolus? Well, I've been telling you it mediates phase separation. So, so here you see what phase separation looks like in vitro. So in this experiment, nucleophosmin was mixed with a single ribosomal protein, RPL5. And you can see that these droplets fuse rapidly. The components within them are highly dynamic, and the droplets are dense and viscous. So how does nucleophosmin do this? So its structure is illustrated here. So at its N-terminus is a oligomerization domain illustrated in the upper right, which is a pentameric hub for that creates extensive multivalency of the other domains in the protein. At the C-terminus is a small helical nucleic acid binding domain, which binds to certain DNA structures as well as to ribosomal RNA. And then finally, connecting the two folded domains is a 120 residue long intrinsically disordered region, or IDR, that contains two stretches or tracks of acidic residues that we call A2 and A3. So we have in total 10 of these acidic tracks in this disordered region in the pentameric nucleophosmin. And these acidic tracks mediate electrostatic interactions with clusters of arginine residues in other proteins that we call arginine-rich motifs. So shown here is a charge density plot for the IDR of nucleophosmin, and you can see the intense negative charge associated with A2 and A3. And so these acidic tracks then interact weakly and transiently with clusters of arginine residues or Arg-rich motifs in other proteins. And so the pattern of Arg motifs in RPL5 that you saw the phase separation of just a moment ago with nucleophosmin are illustrated here. And so you can think of these so this is a disordered polypeptide before entering the ribosome. And so these clusters of arginine residues interact with these acidic tracks, and they form crosslinks or bridges between different pentamers, forming the networks of interactions that I illustrated in my introductory slide. So that's how nucleophosmin mediates phase separation with proteins. But I also told you that nucleophosmin phase separates with ribosomal RNA. This is an example of heterotypic phase separation and is schematized here with the RNA molecules establishing crosslinks, non-covalent crosslinks between nucleophosmin pentamers. And then I just discussed phase separation with ribosomal protein, and nucleophosmin phase separates with dozens and dozens of nucleolar proteins, either ribosomal or non-ribosomal, that exhibit multiple arginine motifs. And this is a second example of heterotypic phase separation, and it's schematized here. Finally, a couple of years ago, we discovered that nucleophosmin by itself can undergo homotypic phase separation under crowded conditions, as of course exists in the nucleolus. So nucleophosmin is really quite remarkable in being able to phase separate through three different mechanisms. So how did these mechanisms contribute to ribosome biogenesis? So a couple of years ago, we published a molecular handoff model of ribosome biogenesis in the nucleolus. And more recently, we put this model to a test together with Josh Reebok and Cliff Brangwyn and their co-workers at Princeton, along with a couple of current and a past member of my lab. And so our model for how NPM directs the vectorial ribosome biogenesis in the nucleolus is illustrated in the center of the slide. So at the top is a mini nucleolus with its 3 layers. And so remember that the ribosomal RNA is transcribed in the interior, and then it's processed, and then it fluxes outwards into the granular component, which is expanded below. So what we envision is that the ribosomal RNA, as it emerges into the granular component, instantaneously phase separates with nucleophosmin. But due to the constant transcription of the RNA, it moves outwards. At the same time, ribosomal proteins enter the nucleoplasm and then are sequestered through phase separation with nucleophosmin. So these blue squiggly lines are the ribosomal proteins. But there's a concentration gradient from outside in, and so those proteins move inwards, opposite to the flux of the RNA. So somewhere in the interior, then, the ribosomal RNAs and the ribosomal proteins will interact and co-fold into ribosomal subunits, and their co-folding then breaks their interactions with nucleophosmin. So that then allows for assembled ribosomal subunits, which no longer interact favorably with nucleophosmin, to passively diffuse out of the nucleolus. And so in the recent study with Cliff Brangwyn and his co-workers, we demonstrated that nascent ribosomal RNA, so in this case 16S nascent RNA, favorably partitions into nucleophosmin condensates. So here is a nucleophosmin condensate into which this RNA has partitioned through phase separation. In contrast, the same RNA, but packaged with ribosomal proteins into the 30S subunit, really fails to partition, or partitions very little, into the nucleophosmin condensates. And this is quantified below with the Gibbs free energy of transfer, with the large negative values for the nascent RNA corresponding to highly favorable partitioning into the nucleophosmin condensate, and these values showing unfavorable partitioning. So this behavior was shown a second way. In this case, the nascent ribosomal RNA was mixed together with nucleophosmin, and the 2 biomolecules underwent phase separation to form the condensates illustrated here. And this behavior is quantified below. So as nucleophosmin concentration is increased, then phase separation is enhanced, whereas very little phase separation occurs with the pre-assembled ribosomal subunit. So this shows that our model is accurate in that ribosomal RNA readily phase separates with nucleophosmin, but as ribosomal subunits assemble, they essentially bury their interaction sites for nucleophosmin and then can passively diffuse out of the nucleolus, as illustrated here. So to sort of summarize and conclude this section of my lecture, phase separation enables the compartmentalization of complex biomolecular processes, such as ribosome biogenesis within the nucleolus. I mean, it's really amazing how it's as simple as differences in surface tension between the 2 layers that is responsible for this compartmentalization. So the evolution of proteins with different phase separation properties, i.e. Different surface tensions, then provides the ability to create multi-layer condensates that then can partition different aspects of biomolecular processes within those different compartments. And finally, multi-component phase separation, which is what occurs within the granular component, provides a mechanism for the biomolecular composition of condensates to dynamically adapt to changing cellular conditions and needs. So an example of this is something observed in this recent study is that so cells expressing different total concentrations of nucleophosmin then display nucleoli with different concentrations of nucleophosmin within them. So there's an almost 40-fold difference in the nucleolar concentration of nucleophosmin going from left to right in these images. And this variation in the concentration of nucleophosmin within nucleoli is quantified in the graph. And what's important about this is that this allows cells to respond dynamically to the needs of the cell. So mTOR signaling drives the production of ribosomal components, but it also drives the expression of nucleophosmin. So as the cell needs more proteins, it needs to gear up the protein synthesis machinery, then the cell responds by increasing the concentration of nucleophosmin in nucleoli, possibly to enhance the capacity of nucleoli to produce ribosomes. So phase separation allows for this sort of complex adaptability and to adaptively control biomolecular processes. So before I conclude, I just want to briefly tell you about a couple of ongoing studies. So in one project, we seek to recreate the nucleolar granular components in the test tube and to recreate ribosomal subunit assembly in these in vitro conditions. And some of our initial results in this area are illustrated on the left. And so we've mixed together nucleolar proteins and ribosomal components and observed that these condensates decompose into multiple phases. And we think that this reflects the granularity observed by electron microscopy years and years ago within the granular components of the nucleolus. And so this is an area of research that we're pursuing very aggressively to basically assemble ribosomes under phase separated conditions in the test tube. And what's really cool is you can manipulate the relative concentrations of the different components to create really interesting looking multi-phase condensates. And then in a second project, we're studying the ARF tumor suppressor protein, and we seek to understand the role of phase separation by ARF in the nucleolus in oncogenic stress responses and tumor suppression. And so nucleophosmin, our results show, immobilizes nucleolar components, including ribosomal components, and inhibits, and others have shown in the past that ARF inhibits ribosome biogenesis. And so we think that this occurs through the immobilization of the phase-separated components of the nucleolus. And I hope to be able to report the results of these studies sometime in the future. And so here I can take a break if there are some questions that have accumulated.
Oh, lots of questions, Richard, and it's absolutely fantastic. I apologize in advance if I mispronounce your name. I will go through as many of these as we can in a short time in interest of the seminar time, but I will get to every question in order after the seminar if I don't reach your question, so please be patient. These are great questions, everybody. Your first question is from Madan Mohan, who asks, can you please tell us what determines the differences in surface tension between droplets made of Fib1 and NPM1?
Yeah, so those differences are rooted in differences in the strengths of the interactions between the different biomolecules. I didn't have time to go into these details, but in the paper we published together with Rohit Bapu and Cliff Brangwyn, we showed that fibrillary phase separates at much lower concentrations of those two biomolecules than does nucleophosmin. And so that means that the strength of the non-covalent cross-links between fibrillary and ribosomal RNA is greater than those the interactions driving phase separation with nucleophosmin. And in addition, fibrillary is a more hydrophobic protein than nucleophosmin. So the fibrillary droplets have a hydrophobic character, whereas the nucleophosmin ribosomal RNA droplets have a highly hydrophilic character. So the differences in surface tension are rooted in differences in the nature of the intermolecular interactions that underlie phase separation.
Barthélemy Diouf asks, what happens if a cell does not express Fib1? Do you still have phase separation between DFC and GC? Are there other proteins besides Fib1 implicated in DFC function in ribosomal RNA processing?
Yeah, that's a really good question. And I actually don't know that there's data on a Fib1 knockout. My suspicion is so we know that knockout of nucleophosmin is embryonic lethal, and I would suspect the same for fibrillary, given how sort of important it is in the DFC. So I don't know that there's specific data to directly address that question, but sorry that I can't provide more insight into that.
Richard, I'll ask one more question, which is actually from two different people. Everyone else, I promise to get to your questions when we finish. Rachiata Darmat and Charlie Roberts both ask if there are specific parameters of ribosomal RNA that favor the interaction with NPM1 as opposed to, say, nascent tRNA or mRNAs.
Yeah, that's a really important question, and is a focus of our ongoing studies. So there really is, for example, no structural data yet on how nucleophosmin specifically how it interacts with ribosomal RNA. We do know that it interacts very favorably with nascent ribosomal RNA and less favorably with partially folded ribosomal RNA. So we suspect that it interacts probably with certain types of secondary structures that are then sort of become buried in the context of the assembling ribosomal subunits. So the detailed answer to that question has to await our further studies, but that is a major focus of our continuing research.
Several more terrific questions, but we'll get to them at the end.
All right, so I will continue and change gears entirely to the topic of phase separation by NUP98 fusion oncoproteins in leukemia. And so the people who contributed to the specific study, the specific results that I'll present in the next slides are shown here. Michael White and Deanna Mitria are past lab members, as also is Jingjing Chen, a former rotation student, and also Brittany Kiosko, current grad student, and Bapa Chandra have also contributed, and we also are collaborating with Scott Blanchard at St. Jude on single molecule fluorescence studies, and Charles Mulligan and his co-workers and the cancer biology aspects of what I'll discuss. So chromosomes can occasionally break and fuse. So these chromosomal translocations create fusion proteins that are very often drivers in cancer, and we refer to these as fusion oncoproteins. A very incomplete list of such fusion oncoproteins is provided here. Fusion oncoproteins are seen in leukemias in particular, but also in solid tumors, and hundreds of them have been described. In particular, we're interested in the NUP98 fusion proteins, and these are seen in AML and in particular in pediatric AML as well as other pediatric leukemias. Fusion oncoproteins exhibit gain of function through rewiring of interaction networks. And we hypothesize that this network rewiring is often mediated through phase separation by fusion oncoproteins. Fusion oncoproteins often fuse a disordered region of one protein to a folded chromatin binding domain of another protein, creating what we think of as aberrant transcription factors. So, as I said, the NUP98 fusions are driver oncogenes in pediatric AML and other leukemias. And so there are more than 30 different fusions, NUP98 fusions, that have been described, and the associated diseases are listed on the right. So these oncogenes fuse the disordered N-terminal phenylalanine glycine-rich domain of NUP98 to folded C-terminal domains of transcription factors, epigenetic regulators, and other proteins that have something to do with chromatin biology. And so we see here at the top of the list the Hox family transcription factors that are fused to NUP98. There are epigenetic regulators, writers, and erasers, as well as other types of proteins that are involved in these fusions. So the NUP98 fusion oncoproteins are known from work of others to drive aberrant gene expression and transform hematopoietic cells. So in particular, one that we have been studying is the fusion of NUP98 with HOXA9, which will be abbreviated NHX9 in my slides. So NUP98 is a nucleoporin that forms the permeability barrier of the nuclear pore complex. So the C-terminal region anchors NUP98 to the ring, the structured ring of the nuclear pore, and the N-terminal domain, which is rich in phenylalanines and glycines, fills the pore center and forms a gel-like barrier to biomolecular diffusion. So it's the N-terminal, the disordered N-terminal FG-rich domain of NUP98 that is fused together with other domains in these fusion oncoproteins. Almost 15 years ago, it was shown by Dirk Gorlich that this N-terminal domain of NUP98 forms hydrogels in vitro. And so the polypeptide, the disordered polypeptide chain forms intermolecular crosslinks that form the hydrogel shown here. And while phase separation wasn't proposed as a mechanism for this back then, this probably occurs through phase separation. HOXA9 is a transcription factor that regulates cell growth and differentiation. It has at its C-terminus a DNA binding homeodomain that binds specific sites in DNA and positions the transcription factor to then regulate gene expression with the Hox family genes and other hematopoietic cell developmental regulators as the targets of this gene activation. So it's this C-terminal folded DNA binding domain of HOXA9 that's fused with NUP98. So that's shown schematically here. So again, this is the disordered FG-rich domain of NUP98. It's a very sticky domain, readily undergoes intermolecular interactions, and this is the folded sequence-specific DNA binding domain of HOXA9. So this fusion protein drives formation of condensates in vitro, as shown in the lower left. So it forms these sort of clumped together, very small condensates, and that's due to the high affinity of the intermolecular interactions that are driving this biomolecular condensation. In addition, this fusion protein forms chromatin-associated puncta in cells, as illustrated here. And at higher resolution, you can really appreciate the sort of punctate, round nature of these condensates in cells. And so you see that there are hundreds of these in the nucleus of this cell. And so we hypothesize that these are binding chromatin and undergoing phase separation to form these punctate condensates. So others have shown that the NUP98-HOXA9 fusion protein drives aberrant gene expression that then transforms hematopoietic cells. And based on recent work of many others, including Rick Young and his colleagues, as well as Xavier Darjats and Robert Tijan and others, we hypothesize that this fusion protein, that the puncta formed by this fusion protein are aberrant transcription centers that form through liquid-liquid phase separation. And so this concept, or this idea, is illustrated in the lower right. So this yellow and green graphic is the fusion protein, which binds to specific sites in DNA, and it can bind to multiple such sites. And then through phase separation of its N-terminal NUP98 domain, it forms this type of transcription center or transcriptional condensate. We propose that these transcription centers recruit components of the transcriptional machinery, including co-regulators, polymerase II, et cetera, via co-phase separation. So these other components of the transcriptional machinery are represented by the different colored icons in this image. And when the right constellation of components are present, then gene expression can be activated. So to probe the causal role of phase separation in fusion protein-driven gene expression, we truncated the full-length construct to this construct that we've referred to as NHX9 MIDI, which our results show undergoes phase separation like the full-length protein, both in vitro and in cells. So then this, so very often, structural biologists like to take a minimalist approach to understanding functional mechanisms. And so we were using this MIDI construct as a template for mutagenesis to modulate phase separation and to test whether this alters gene expression. So, so progressive mutation of phenylalanines and glycines in this MIDI construct reduced phase separation in vitro. So we mutated a small number and then a large number of these phenylalanines, and in some cases phenylalanines and glycines, and the in vitro phase separation results are shown below. So these are the results of a turbidity assay, which can be used to monitor the formation of micron scale condensates in solution. So the wild type MIDI construct forms condensates in a concentration dependent manner. The construct with 6 or 8 phenylalanines mutated to alanine undergoes moderate phase separation, but the more extensively mutated constructs do not undergo phase separation at all. So this variation of phase separation behavior is also observed in cells. So again, the wild type construct sort of causes the formation of hundreds of small puncta in cells, as illustrated here in the left. But the two of the mutated constructs show diffuse staining pattern. There are some puncta in the central image. It's probably very hard for the audience to see that, but there are no such puncta in the most extensively mutated construct. So we've begun to probe the gene regulation features of these mutated constructs, and our early results are illustrated here. So we used qPCR to monitor expression of HOXA9. So HOXA9 is a hallmark gene of NUP98 fusion protein transformed cells. So the HOX gene cluster is pretty much always upregulated in AMLs driven by NUP98 fusion oncoproteins. And so we see that the full-length fusion protein causes significantly increased HOXA9 expression, as does the wild-type sequence MIDI construct, although to a lesser extent. But the two phenylalanine mutants exhibit no gene upregulation at all, nor does a mutant in which the homeodomain is mutated to not bind DNA specifically. So these results show that phase separation is correlated with gene expression in this simple cell assay. And collaborative studies that are right now ongoing with Charles Mulligan and his postdoc Nicole Mitchmer-Hughes in pathology at St. Jude show that the MIDI construct transforms hematopoietic stem cells, so the cell of origin for AMLs driven by these fusion proteins, similar to the full-length construct. So this means that this MIDI construct and the mutants that I've just discussed can be used in the future to test the causal role of phase separation in leukemogenesis in vivo. And we're really excited to carry this work forward in the future. And these studies are actively ongoing between our two labs. So while we have beginnings of an understanding of the role of phase separation in the aberrant gene expression and transformation of hematopoietic cells, there are many open questions. So some of those are listed here. So one key question is, what transcriptional components are recruited to these condensates, and how are they recruited? And is recruitment specific? Another question is, does phase separation by the NHX9 fusion protein actually alter chromatin structure and dynamics, as I've been illustrating schematically? So we've begun tests, experiments to address this latter question. So I'd like to share those results with you. So fusion oncoprotein induced phase separation and DNA compaction then provides a mechanism for bringing enhancer and promoter elements together in space for coordinated gene regulation, as illustrated in this little scheme. So we're using single molecule fluorescence methods, specifically FRET using TURF, to determine whether NHX9 causes DNA compaction. So the experimental design is as follows. We have an about 400 base pair double-stranded DNA that has a HOX binding site at either end. And the hypothesis is that when the fusion protein binds, it causes phase separation together with the DNA, and that brings the ends of the DNA together. And so we've constructed a DNA with different fluorescent dyes at two ends, and have tethered these DNAs to the surface of a glass slide, and you put this in a TURF microscope, and then you can monitor the fluorescence of individual DNA molecules as the fusion protein is added. And we wanted to know whether the ends of the DNA come together through phase separation. So we have done this with so far with two constructs. One is the wild type MIDI construct, and the other is the DNA binding mutants that I described just a moment ago. So this binds DNA only non-specifically. And so very quickly, in the interest of time, so FRET efficiency is on the y-axis, and time after addition of the protein to the TERF cell is on the x-axis. And so when the protein is added, we then see a sharp rise in FRET. So these are the collective results of observation of hundreds and hundreds of single molecules on the TERF slide surface. And the results for one such molecule are shown here. So as the protein is added, we immediately see photons corresponding to FRET, the red photons, and the FRET efficiency is given here. In contrast, the DNA mutant of this construct shows very weak and transient FRET, and that transience is illustrated in these results for a single DNA molecule here. I have to emphasize that this is work that's being done in collaboration with Scott Blanchard and his co-workers at St. Jude. So these results provide a foundation for testing the role of phase separation in DNA compaction using phase separation deficient mutants of this construct in the future. And we'll perform conceptually similar experiments in cells, for example, using single molecule fish to track Hox enhancer and promoter, the proximity of Hox enhancers and promoters in cells to hopefully causally relate phase separation of this fusion protein with DNA compaction, which is something that's yet to be done by others in the phase separation field. So to wrap up now, so transcription factors, coactivators, and RNA polymerase II have intrinsically disordered regions that promote phase separation. And this is work that's been done by Rick Young and others. And this image is taken from one of Rick Young's fairly recent papers. And phase separation organizes the transcriptional machinery to drive gene expression. So all these different regulators, transcription factors, co-regulators, and also polymerase II shown here, which has an IDR that basically co-phase separate and drive gene expression. So we've performed sequence analyses of thousands of fusion oncoproteins and have noted that many have IDRs that we predict promote phase separation, including the NUP98 fusions. And we hypothesize that NUP98 fusion oncoproteins are aberrant transcription factors that drive aberrant gene expression through phase separation. So essentially, here's a NUP98 fusion oncoprotein. It can bind specific DNA sites, and its N-terminal region can phase separate. And we propose that this sort of catalyzes the formation of aberrant transcription centers that are responsible for driving the aberrant transcriptional program known to be associated with these fusions and AML. So the big question, our big challenge is to prove that these puncta are in fact phase-separated transcription factors that are driving aberrant gene expression. So just to bring the different parts of the talk together quickly. So phase separation by fibrillary and nucleophosmin with ribosomal components creates the multiple layers of the nucleolus. It's really just amazing how it's as simple as fluid physics. And this organizes the different steps of ribosome biogenesis, including the transcription of ribosomal RNA and its processing, as well as subunit assembly in the respective layers. The multiplicity of interaction mechanisms and high multivalency of nucleophosmin, illustrated here, enable multiple modes of phase separation that facilitate ribosome subunit assembly in the granular component. And so we see in studies of the nucleolus then many different ways that phase separation contributes to the important function ribosome biogenesis. And then with fusion proteins, so fusion oncogenes often encode fusion oncoproteins that we hypothesize phase separate to form transcription centers that drive aberrant gene expression and transform cells. So we're super excited about both sort of areas of study and look forward to exploring sort of additional aspects of how phase separation contributes to normal function in the case of Nucleolus and aberrant function associated with cancer in the case of the second. And so with that, I would again like to thank and acknowledge coworkers, and we'll be happy to take any questions at this time. So thank you very much for your attention, and yes, happy to take questions.
Thank you so much, Richard. Outstanding work, really exciting, and there are a great many questions. Officially, we're at the top of the hour, so we understand that some people have other things scheduled now and may have to leave. But at this point, we'll start our open discussion for 30 minutes. Still, send your questions and comments to the chat, and I'll bring them up in order as they go. I'm also going to use my prerogative as chair to ask perhaps the first question. Richard, on the nucleolar part that you presented to us, we've known for many decades that as cells enter mitosis, the nucleolus essentially dissolves and reforms at the end of telophase. Presumably, this is all coordinated through cyclodependent kinases and phosphatases. Do you have any idea at all about how modifications of NPM1 or FIB1 by these machineries might control phase transition?
So we definitely have some insights into that. We published an early paper on nucleophosmin prior to our phase separation studies that showed that phosphorylation of certain residues within the oligomerization domain disrupt the pentamer and essentially cause the pentamer to unfold into disordered monomers. And some of those phosphorylation sites are sites for cyclin-dependent kinases. And so what's known phenomenologically is that the granular component dissolves during mitosis, and that the inner subcompartments sort of break up into small diffusible bodies. And so we hypothesize that nucleophosmin is being phosphorylated, its pentameric structure is disrupted, which reduces multivalence and therefore will disrupt phase separation. So we know that monomeric forms of nucleophosmin undergo phase separation only at very, very high concentrations with ribosomal components. And so therefore, so that's what we think is occurring when the nucleolus, the outer region of the nucleolus, dissolves during mitosis.
Fantastic. A graduate student from Rutgers named Aishwarya Ranganath asks whether NPM1 engages through K-rich motifs, lysine-rich motifs. Does it also, or are R-rich motifs also relevant?
In earlier studies, we showed that a common feature of many nucleolar proteins, I think at least half of the nucleolar proteins fit this feature, and that is that they display multiple arginine-rich motifs. And so we think that arginines in particular are important for interactions with and phase separation with nucleophosmin. And in some cases, we've mutated arginines to lysines and showed that this diminishes or reduces phase separation. So arginines seem to be particularly important for driving phase separation with nucleophosmin. Not to say that lysines don't contribute at all, but the chemical nature of the primary amine of lysine and the guanidinium group of arginine are very different, and they mediate electrostatic interactions and other other types of interactions differently. So so arginine-rich motifs are particularly important in interactions and phase separation with nucleophosmin.
Alex Kensis and Chuck Scheer both asked very similar questions about the NPM1C mutations in AML that drives NPM1 to the cytosol. Any idea what happens in such cells?
So our studies have been very limited because since many of our studies are in vitro and the C mutants are misbehaved proteins due to the C mutations that are associated with AML disrupt the structure of the C-terminal RNA binding domain. And so in our past studies, we showed that that domain was critical for the nucleolar localization of nucleophosmin. So when the domain is unfolded by the mutations, can no longer interact with ribosomal RNA, and that probably explains its cytosolic localization. So that may, so if if half the nucleophosmin molecules in a cell are mutated and localized in the cytosol, that may alter nucleophosmin's role in the nucleolus, possibly. Although Scott Armstrong showed recently that these C mutants alter gene expression in the nucleus, and nucleophosmin is known to interact with chromatin. In addition to being localized in the nucleolus, it also does interact with chromatin and have some roles, although they're poorly understood. So it's possible that there are effects of the C mutants in the nucleus, in addition to the protein being heavily localized within the cytosol. But I'm sorry I can't provide a more clear answer to that question, but it's a very important question, and we hope to address it in the future.
Danfeng Kai says, awesome talk, and I think we all concur with that. Your NHX9 expressed in cells looks more like a network than spherical condensates. Do you have any idea if the endogenous NHX9 also forms these network-like structures, and if so, does that have physiological relevance?
So I'm not sure what's meant by endogenous NHX.
I guess in a cell, I guess.
Okay, so maybe they're referring to the appearance of the NHX9 condensates. In vitro, they appear as sort of clumps of very small condensates. So we think that the condensates initially formed through liquid phase separation, but they rapidly undergo a gel transition and then sort of clump together, as was evident in vitro micrographs. And then in cells, we've done limited studies of the material properties of the NHX9 puncta in cells, and FRAP of the TFP NHX9 shows that those molecules are quite dynamic, and maybe didn't display as clearly as it looks on my screen, but the high res image of those puncta that I showed in the beginning part of that part of the talk really appear to be quite round and quite dispersed through the nucleus. So we think that we really feel quite confident that those are sort of liquid-like bodies that probably are incorporating many of the transcriptional components, but that's the subject of our ongoing studies.
Great. Jinwei Cao has a fairly specific question. In the transcription activity assay you showed, the qPCR of HOX9, did you compare ectopically expressed NHX9 to ectopically expressed HOX9?
No, we haven't made that comparison yet, and that's something that we obviously need to do in the future.
Madan has another question. Madan Mohan, in NUP91 fusion cancer cells, does one observe reduction in the number of nuclear pores or global imbalances in nucleocytoplasmic partitioning of nuclear proteins such as transcription factors?
So it is known that the NUP98 fusions do impact transport, nuclear cytosolic nuclear transport, and I'm not sure if it's known that transcription factors in particular are impacted by that, but it is known. Under certain conditions, you see the fusion proteins localized not only in nuclear puncta, but also at nuclear pores, and they do have effects on transport through the NPC.
This is relevant. By the way, Madan also added, great talk, Richard. I concur. I'm going to skip to James Dennis, and I'll come back to the two questions before him because it's the same subject. NUP98 as a hydrogel in nuclear pores forms a barrier which is loosened by O-Glicnak modification of multiple serines in the FG regions to allow cargo transport. Do you think this might have a role in the fusion oncoprotein story?
That's a really interesting point. And honestly, not something, I mean, I'm familiar with this modulation of sort of nuclear pore behavior by this particular modification, but actually haven't considered it in the context of the NUP98 fusion oncoproteins and the condensates they form in nuclei. That's a very good suggestion. What I will say relative to nuclear pore behavior, it's known that CRIM1, a nuclear exportant, does in fact localize to NUP98 fusion oncoprotein nuclear puncta.
Oh, interesting.
Yeah, and we're currently making CRIM1 to study its interactions with condensates in vitro and also in cells, and so there may be, it's very likely that there's some importance to so basically the aberrant condensates with fusion proteins may be sort of mislocalizing CRIM1 and possibly CRIM1 cargos. So yeah, that's a very interesting point. Thank you for that.
Stephen asks, from the FRET efficiencies, what is the distance between the DNA ends in the presence of NHX9 using your SM FRET technique?
Yeah, that's a good question. We actually haven't analyzed the data quantitatively with respect to interdye distance yet, so I can't say specifically. The dyes are are attached with relatively short linkers. So, I think, from my recollection of sort of the range of distances probed by the particular dye pair, distances probably have to be something in the range of maybe 30 to 40 angstroms, I think, or maybe, yeah. So that's the best I can say right now.
There's, let's see, two comments from Anirban Roy Chowdhury, and I'll put those together. He says there are over 150 other nuclear proteins. Do these also function to maintain the tripart structure of the nucleolus, or what is the role of these clients in a condensate, noting that these clients don't take part in ribosomal synthesis?
Yeah, so yeah, that's a very good question. And so, I mean, our studies have focused on just a few components of the nucleolus. And so fibrillary and nucleophosmin are highly abundant proteins in the nucleolus. And we've shown that the behavior of these two proteins in vitro explains some critical features of nucleoli. But we totally acknowledge that there are hundreds of other proteins present that are obviously there to contribute to ribosome biogenesis. And but we don't yet really understand how different proteins contribute in different ways. I know that in the dense fibrillar component there are sort of many different types of enzymes that splice the ribosomal RNA. There are various SNO RNAs that guide the specificity of those reactions. Fibrillary in itself is a methyltransferase and binds to SNO RNAs that direct it to particular sites within ribosomal RNA to methylate bases. Pseudo-uridinylation is carried out, but the extent to which these other proteins contribute to the material properties of the different regions of the nucleolus, we don't yet understand. But it's an important question that just will require further study in the future. I mean, it depends on who's counting, but an important proteomic study in I guess about a decade ago reported that there were almost 800 proteins in the nucleolus. And then a study conducted by this was Lamon's group with Angus Lamon with Matthias Mon, and then Angus Lamon himself. I guess he got a more sensitive mass spectrometer, and then he counted several thousand proteins in the nucleolus. So obviously there are other proteins contributing to the features of nucleoli, but I hope that I've convinced people in the audience that the behavior of just a few can explain many of the sort of key and defining features of nucleoli.
Finally, one last question from me, Richard, unless any come in over the next few minutes. You very quickly alluded to P19-ARF and its effects on ribosomal biogenesis through interactions with NPM1. Does P19 disrupt the phase transition, or from another point of view, are there cancer-associated mutations in NPM1 that make the phase transition induced by NPM1 resistant to any effect of P19-ARF?
I think the answer, well, we haven't studied any of the cancer-associated mutants of ARF. I think more frequently, ARF is silenced in cancer.
Oh, I'm sorry. I was talking about the NPM1 mutations.
Oh, the NPM1 mutations and interactions with ARF. Well, certainly, yeah, that's and I don't know if if silencing of ARF is required in those AMLs. Um, so so let's see. So ARF would still be able to interact with C mutant NPM, but I don't know, and it's possible that that could cause mislocalization of ARF out of the nucleus because the C mutants are cytosolic. So that is an aspect of the NPM C mutants that we haven't actually given very much thought to, but it's a very interesting question, Doug.
Yeah, I think there's a lot to unpack there. Let me just check to see if we have any more. Madonna is still here and asked one more question. NHX9 fusion protein, since in the NX9 fusion protein, since the activation domain is removed by the DNA binding domain and the DNA binding domain is retained, is it likely that the aberrant condensates may represent transcriptionally inactive repression centers, i.e., is there a dominant negative effect antagonizing normal HOX9 activity?
So I don't know that there's support for that mechanism. So what's known is that binding to specific sites in chromatin is required, and for aberrant gene expression. And if you either mutate the chromatin binding domain so that it doesn't do that, or you chop it off, then you see the formation of large nuclear puncta that are not chromatin associated. So I would think that if it's sort of dominant negative mechanism that and those chromatin those chromatin displaced puncta do not alter gene expression. So I would think that if it was a dominant negative mechanism, that those non-chromatin associated puncta might also still have those dominant effects. So we hope to directly address this question in the future by doing live cell imaging of the transcribed RNA at a particular enhancer promoter site. Of course, it's a formal possibility, but our current thinking is actually opposite that. But it's important to consider all possible mechanisms, and we certainly will keep an open mind as we go forward with our studies.
That's wonderful. Let me see there. Okay, we've got a few more questions just came in at the last minute. Charlie Roberts asked, is it known whether ribosomal RNAs are the only chromatin regions that localize to the nucleolus, or might other regions and their RNA contribute as well?
That's a really good question. I think the answer is that it's pretty much just the tandem repeats of the ribosomal RNA genes. So these are found within the short arms of 5 acrocentric chromosomes in humans, and those chromosome regions they actually have a very unusual structure. So they appear in EM images as being diffuse and almost as like breaks in those short arms. And that's due to the binding of a specific set of proteins to some of the DNA elements in those tandem repeats. I think it's pretty much just these tandem repeat regions of these particular chromosomes that sort of are threaded through nucleoli, and essentially these gene regions give rise to nucleoli because when RNA pol 1 is active and it binds to these regions, then the ribosomal RNA is produced, and that's sort of the origin of a nucleolus. There are nucleoli do become associated with other chromosomal compartments. So nucleoli can, under certain conditions, can form caps, and I think the periphery of nucleoli interact with other sort of types of chromatin in the nucleus, but I think it's just the tandem repeat regions that are within the interior.
Finally, Madan says, thank you for a great talk, Richard, and all the wonderful insights, and we all concur. We will this off, and thank you for all of those who stuck around, quite a few. And again, thanks for an amazing talk, Richard.
Thank you, Doug, and thanks for the opportunity, everyone, and thanks for attending, and have a good rest of your days.
Thanks, Richard.
Okay, thanks, Doug, for chairing.
My pleasure.